Hello friends, how are you doing? And I hope you all are doing good and exceptionally well. So I hope before sitting for this NEET based question exercise, you are done with the biological, you're done with the theory of the biological classification, right? Okay, so let us solve some questions which are based on the NEET pattern exercise. The first one, which statement is wrong for the viruses? Yes, we know virus infect the body, and virus are virus, they behave. What is one of the important characteristic of viruses? The viruses behave as both living as well as non-living, behave as both living as well as non-living, right? So the viruses behave as both living and non-living, and what will we see here further? Viruses behave as both living and non-living. Now what will we see in these conditions? That yes, the viruses multiply within the body of the host. Viruses multiply within the body of the host, within the body of host, right? Now all are parasites. Which statement is wrong? All are parasites. Yes, almost all of them are parasites. So this is right. So this is not our answer. All of them have helical symmetry. Now this is something to be considered. Not all of them have helical symmetry. Okay, the viruses they have a very compact shape. The viruses, the nucleocapsid, the nucleic capsid coat of the viruses, they are basically either they make the virus either rod shape or spherical shape. The rod shape will be having the helical symmetry, right, and the other one will be, that is the spherical shape, will be having the icosahedral symmetry. So all of them have helical symmetry. This is absolutely wrong statement, and hence this is our answer, or this is the option for this particular question. The next one, they have ability to synthesize nucleic acids, absolutely correct. Antibiotics have no effect on them, absolutely correct. Okay, so we'll just write that down, whatever we discussed right now about viruses. The nucleocapsid coat of viruses, offers a definite structure, to them, right? These viruses are mainly of two shapes, right? So these viruses are mainly of two shapes, right? And what are those two shapes? Rod shaped and spherical shaped, right? So rod shaped and spherical shaped, right? So these viruses are mainly of two shapes, rod shaped and spherical shaped, and we will see the rod shaped viruses, right? The rod shaped viruses have spherical symmetry, right? The rod shaped viruses have spherical symmetry, while the spherical shaped viruses, while the spherical shaped viruses have icosahedral symmetry. Icosahedral symmetry. Right. The rod shaped viruses have spherical symmetry, while the spherical shaped viruses, right, have the icosahedral symmetry, and hence that is why we chose the B option as our answer. Once again, I'll see whether I've written right. The rod shaped viruses have spherical symmetry, while the spherical shaped viruses have icosahedral symmetry, right. So now we'll move on to the next question, right. We'll talk about the next question, and that is some hyperthermophilic organisms that grow in highly acidic habitats, highly acidic, pH very less, like 1 or maybe less than 1, 0.7, 0.8, 1.5. Okay, so some hyperthermophilic organisms that grow in highly acidic habitats belong to the two groups called as, right. Right. So whenever when the kingdom Monera, the kingdom Monera was divided, in the 6 kingdom classification, the kingdom Monera was divided into the archaebacteria, and the eubacteria, right. The archaebacteria and the eubacteria, right, in the 6 kingdom classification, right. Now further in the archaebacteria we will see that there are thermophiles, thermoacidophiles, halophiles and all that, which will be resistant to high temperature. Even in eubacteria there are thermophilic organisms which can be resisting the high temperature. So what do you mean by hyperthermophilic? That hyperthermophilic means thermophilic means temperature resistant, and hyper too much. So these organisms will be able to resist, or will be able to tolerate high temperature conditions. This is what we mean, right. So if we want to frame this answer, what we can write, okay, that the archaebacteria, right, the archaebacteria and even some of eubacteria can tolerate high range of temperature conditions. Okay, so they can tolerate high range of temperature conditions. Right. So the archaebacteria and even some of the eubacteria can tolerate high range of temperature conditions. Okay, like for example 60 to 70 degrees centigrade, and even high salinity, and even conditions, okay, like high salinity, right, and even conditions like high salinity as well as as well as high acidic conditions, okay, as well as extreme low and high pH conditions. Right. So, the archaebacteria and even some of the eubacteria can tolerate high range of temperature conditions, like for example 60 to 70 degrees centigrade, and even conditions like high salinity as well as extreme low and high temperature conditions. And that is what we say when we when we want to say that yes, the eubacteria and the archaebacteria are the one. So, some hyperthermophilic organisms that grow in highly acidic habitats belong to the two groups called as the archaebacteria and the eubacteria. Right. The third question, moving on to what are interferons? Okay, what are the interferons? The interferons, so now if we talk about the interferons, these are antiviral, these are antiviral glycoproteins. These are antiviral glycoproteins. Okay. So, these are antiviral glycoproteins that are non-antigenic, right. That are non-antigenic, and these will help and are produced, okay, in the virus infected cells, and are produced in the virus infected cells. Virus infected cells, right. These are antiviral glycoproteins that are non-antigenic and are produced in the virus infected cells. Now, what will they do? These interferons, which are the antigenic, which are the the proteins, the antiviral glycoproteins, these interferons will produce enzymes. These interferons will produce enzymes. Okay, these interferons will produce enzymes that will stop the action of the virus and will and will fight against the action of the virus will produce enzymes that will fight against, okay, that will fight against the action of virus. The interferons will produce enzymes. The interferons will produce enzymes that will fight against the action of the virus. So, the interferons are what? These are the antiviral proteins, right. The interferons are the antiviral proteins. Great. Let's talk about the next question now. Temperature tolerance of thermal blue-green algae is due to, right. Now, the temperature tolerance of blue-green algae, which is also called as the cyanobacteria, is due to what? Basically, this is due to the protoplasm structure, the bonding in the protoplasm, okay, and also the sheath, the sheath present over here, that also contributes to the temperature tolerance of these cyanobacteria, right. The temperature tolerance of cyanobacteria the temperature tolerance of cyanobacteria is due to the sheath, that is the cell wall structure, right. So, the temperature tolerance of cyanobacteria is due to the sheath, that is the cell wall structure, as well as the bonding in the protoplasm. Okay, as well as the bonding in the protoplasm. So, the temperature tolerance in the cyanobacteria, is due to the sheath, that is the cell wall structure, as well as the bonding in the protoplasm. So, that is what we say, the cell wall structure is one major reason why it is there. Temperature tolerance, right. We move on to the next question. African sleeping sickness is due to, right, Plasmodium vivax transmitted by the tsetse fly, Trypanosoma leuci transmitted by the bedbug, Trypanosoma gambins transmitted by Glossina palpalis. Yes, this is the answer, Trypanosoma gambins, which is transmitted by the Glossina palpalis, and this Glossina palpalis is also known as the tsetse fly. Okay. The Glossina palpalis is also called the tsetse fly, which transmits the Trypanosoma gambins, and which is the cause for the African sleeping sickness. Right. So, now we move on to the next question. The infective state of malarial parasite Plasmodium that enters the human body is what? The infective state of the malarial parasite Plasmodium of malarial, okay, the infective state of malarial parasite Plasmodium is the sporozoite, okay, is the sporozoite, right, which is a spherical cell, right. So, the infective state of malarial parasite Plasmodium is the sporozoite, which is which is a spherical cell, and what happens, or which is a spherical structure, we can say organism, that is present in the salivary fluid of the mosquito, okay, Plasmodium that is present in the salivary fluid of the mosquito, right. So what happens, when the mosquito bites, when the mosquito bites, this sporozoite, along with this sporozoite, along with the saliva, gets transmitted to the human blood, from where it catches the infection. So hence on biting, the saliva right, hence on biting the saliva of mosquito, along with, right, hence on biting the saliva of mosquito along with the sporozoite, enters, okay, along with the sporozoite enters the human blood, So the infective state of malarial parasite Plasmodium enters the human body, is the sporozoite, right. Great. Okay. Let's talk about the next question now. During the formation of bread, it becomes porous due to the release of CO2 by the action of, right. During the fermentation of yeast, what happens, the yeast fermentation, what we see, there is liberation of carbon dioxide as well as ethanol, right. And what happens, this carbon dioxide gets released, and because of which, the bread becomes porous. And this happens due to the enzyme, what, zymase. During the fermentation of yeast that happens, okay, that happens in the presence of enzyme zymase, that happens in the presence of enzyme zymase, right. So during the fermentation of yeast that happens in the presence of enzyme zymase, there is released what? Carbon dioxide and ethanol, right. The carbon dioxide gets okay, gets released into the environment, okay. So the carbon dioxide gets released into the environment, thus breaking, thus leaving the bread porous, right. So the answer for this will be yeast, right. The answer for this will be yeast. Let us go to the next question. In fungi, the stored food material is what? In most of the animals, including including human, we see that carbohydrate or glucose is stored in the form of glycogen. So the answer for this will be glycogen. That in fungi, the stored food material is stored in the form of glycogen, right? Yeast, Saccharomyces cerevisiae, is used in the industrial production of what, right? As I told you, the fermentation of yeast in the presence of enzyme will give you what? The fermentation, right? The fermentation of yeast, that is Saccharomyces cerevisiae, in the presence of enzyme, in presence of zymase, right? The fermentation of yeast in presence of zymase forms what? Ethanol. Ethanol is nothing but ethyl alcohol and CO2. Okay, forms ethanol and CO2, and this is what we will see here, right? Ethanol and CO2. Yeast Saccharomyces cerevisiae is used in the industrial production of ethanol, right? And finally, we will move on to the last question. Right? Lichens indicates sulfur dioxide pollution because, now what are lichens? The lichens are, they are symbiotic relationship between the algae and the fungi, right? Lichens are symbionts, okay, who portray or display the symbiotic relationship, are symbionts between the algae and the fungi. Okay. So lichens are symbionts, or we can say lichens are symbionts between the algae and the fungi. Okay, the phycobiont and the two, the phycobiont and the mycobiont. Okay, we can say. Now what we see, here, that these lichens, especially the epiphytic lichens, they are very sensitive. They are very sensitive to pollution and especially sensitive to sulfur dioxide, right? Are sensitive to sulfur dioxide, right? So friends, with this we have come to the end of this session of NEET pattern exercise of this biological classification. Many more questions to follow, but keep on revising and keep on following us. The time we meet again, a very goodbye from this side.